Hello, everyone, and thank you for joining us for our special Thursday webinar today. Just a couple things before we get started. You can use your chat function to send in any questions that you have throughout the presentation, and we'll try to get those answered towards the end of the webinar today. And the certificates will be available for those attendees here in the U.S. on Friday by logging into your account. You can download the certificate. And Nania, if you'd like to introduce our speakers today, go right ahead.
Welcome, everyone. My name is Nania Schade-Hernandez, and I'm the Director of Global Education at Medela Headquarters in Switzerland. Thank you very much for joining us today, and welcome back if you have already been with us for some of the past webinars. We are hosting these webinars because the global COVID-19 pandemic has really made a tremendous impact on the way we work and on the way we function. And we have been greatly concerned in particular about COVID-19 having a negative impact on breastfeeding rates on a global scale. We have seen conflicting information in the popular press and all over the internet about the use of human milk and breastfeeding, even though global organizations like the UN and the WHO have been very clear with their advice that moms can continue to breastfeed. And it is because of these discrepancies that we organized this series of webinars and why we are here today. Really to bring you the world's leading researchers to cut through this information and to provide some clarity on the use of human milk and breastfeeding during this COVID-19 pandemic. Today's installment is the fifth of our series, and we are really fortunate to have Assistant Professor Rebecca Powell with us today. She's an Assistant Professor of Medicine and Infectious Diseases at Mount Sinai's Icahn School of Medicine in New York. When she's not working from home or caring for her 3 kids, including breastfeeding one of them, apparently Dr. Powell finds time to put on a mask and trek across New York City to pick up breast milk from lactating women who have had a positive COVID test. Talk about commitment. Because she studies the immune properties of human milk. Today, she will be outlining the basic immunologic components of human milk, specifically those related to defending breastfed infants and children against infection, as well as talking about the antibody response to infection in human milk and how this differs from the systemic response. And she will also talk about emerging data regarding the antibody response against COVID-19 in human milk. So before I hand over to Dr. Powell, let me remind you to please mute your phones and do feel free to pop some questions into the chat. We should have 15 minutes to address them at the end of the talk. Please welcome Dr. Rebecca Powell.
Hi, thank you very much for that introduction. And so today I'm going to talk to you about our work showing evidence of a strong and specific antibody response against SARS CoV-2 in human milk. I have no conflicts to disclose, and Medela did not have input to the content of this presentation. So as was just stated, the objectives of today's webinar are, to give some background, to learn about the basic immunological components of human milk, to understand the antibody response to infection in human milk, and the general antibody response and how this differs between milk and blood, and to learn about the emerging data that we've generated regarding the antibody response against COVID-19, or specifically the virus that causes COVID-19, which I'll refer to as SARS CoV-2 in human milk. So I'm going to go pretty basic because I know we have a lot of different people online today. So just some background. So antibodies, what are antibodies? An antibody is a protective protein that's produced by the immune system in response to the presence of a foreign substance. And antibodies recognize and latch onto the foreign substance in order to remove them from the body. So this is just a schematic. Obviously, it does not really look like this, but these show the components of the antibody. So you have down here what's called the heavy chain or the constant region. It's also how it's referred to. And then at the other side of the antibody is what would bind to, let's say, the virus, and that is known as the variable region or the Fab region. Because that's what's specifically meant to bind to different pathogens. So that end of the antibody will differ depending on the pathogen it's intended for, and the constant region is relatively constant. That's why it's named like that, no matter what pathogen it is directed at. So when an alien substance enters the body, the immune system is able to recognize it as foreign because its proteins differ from those found in the body. And not all antibodies are created equal. Of course, it's not so simple. It never is. So we have so many different types of antibodies. We've got IgG, which is what you would hear most about as being in the blood. We have other ones, IgM, which is tends to be the first produced, the first measured after infection. And then IgA, we have milk is 90% IgA. So IgA is what's known as the mucosal antibody. Mucosal means sort of those, you know, those wet secretions that are made by our bodies at different sites. You can imagine, you know, saliva, nasal secretions, and human milk, for example. 90% of antibody in milk is of the IgA type. So I'm going to talk mostly about that today. And then there's also IgD, which is very unstudied and not well understood, and IgE, which you might have heard of, which is involved in allergic reactions. So now some background on the antibodies in human milk. So as I just said, human milk antibody is about 90% IgA, and it's also about 8 to 10% IgM. So those are, you know, what you might think of as the most important antibodies in milk. There's still going to be a bit of IgG, maybe the others as well, but very minimal. And nearly all of these antibodies in milk are in what's called secretory form. They're secreted, right? So they're referred to as SIgA or SIgM. And these secretory antibodies are polymeric, meaning they have multiple components, and they're complex to two different proteins in addition to the antibody molecules themselves, which are known as the J chain and secretory component. So I have some diagrams here to illustrate that. So here we can see there's two different kinds of IgA, and I'm not going to get into that today, but just so you know, it's, again, pretty complicated. There's many subtypes of all sorts of proteins in our bodies. But you can see here, again, this schematic of an antibody. You've got your constant region, you've got your variable regions up here. So that would be a monomer, meaning there's just one. Now, in the blood, you will find IgA, but it will just be a monomer, like that. Now, in B cells, which make antibodies, when those are destined to produce secretory antibodies, those cells also make something called J chain. And that's another protein, and this protein helps join two molecules of IgA. So then it's called a dimeric IgA molecule. Now, I'm going to show you how this happens in a minute, but the full secretory IgA molecule has the typically two IgA components, and then it has J chain that holds them together, and then it also gets kind of, you know, printed with secretory component, which is SC as I'll refer to it. And then that is the full dimeric secretory IgA, which is really what is the most important and most common antibody molecule in milk. So as I said, so in humans, milk, it does have some IgG, which is the antibody type that's the most dominant in serum, in blood. Milk has a bit of that, about 2%. And that will come mostly from the serum. It kind of seeps in, if you will, from the blood vessels. B cells that produce SIgA and SIgM, they actually originate from the mucosal-associated lymphoid tissue, or what's called the MALT. And most of that is really going to come from the gut, which is the gut-associated lymphoid tissue, but also with some respiratory mucosa involvement as well. So that means that the cells that end up in the mammary tissue in the breast to produce these antibodies into the milk, those cells actually they travel from other mucosal sites, mainly the gut. So this is known as the entero-mammary link, and this has been pretty well studied, you know, mostly in other mammals, because of course it's easier to do all sorts of studies in animals, but you know, all mammals lactate, and so it seems that all mammals have this entero-mammary link, and a lot of studies been done in mice and some in some other animals as well. So this is illustrated here. This is not my figure, but this is a commonly available figure. So, for example, if a person would ingest a pathogen, obviously not on purpose, just an environmental pathogen or something pathogenic, and then this activates B cells in the gut in what's known as the Peyer's patches, which is sort of like a gathering of immune cells in your small intestine. And then this causes an immune reaction, and then part of that immune reaction is those B cells migrating by a really finely tuned process, by our evolution. They migrate through the lymphatic system, and they make their way to the breast tissue. And then those are the B cells that secrete antibodies specific for what the mom had eaten, for example. Now those antibodies go into the milk, and then they go into the baby's gut and protect the baby from those very same pathogens the baby is also likely to encounter orally, right? So you can imagine why evolutionarily this is quite perfect, and this is the way it was intended. There's also some involvement from the respiratory mucosa as well, so pathogens that would be inhaled, that has been demonstrated. So evidence of this from animal studies is that mammary gland B cells, they show a surface protein profile. The types of proteins on the surfaces of B cells that you can find in the mammary gland match the ones that you find in the gut very well, and they don't match the ones you find in other compartments like the blood. And milk secretory IgA exhibits reactivity for enteric pathogens. So you find all sorts of antibodies in milk that are specifically made to bind to the types of viruses and bacteria you have in your gut. And that's not what you find in other compartments. And then an interesting mouse experiment, I'm not going to get into detail, but these mice were made to have extra long small intestines. And those mice were found to have more B cells in the mammary gland and more secretory IgA in the mammary gland. So that's also very interesting. So this homing of the gut B cells to the mammary gland depends on a lot of special molecules on the surface of cells called adhesion molecules. So they adhere, they're sticky, right? So that's what gets these cells to basically roll along the lymphatic pathway in a particular direction, because it's a very finely tuned system that makes sure that the baby is getting the right kinds of antibodies that originate from the mom's gut and respiratory tract. So once these cells reach the mammary gland, the IgA and IgM are produced, and they bind to something in the mammary gland called the polymeric immunoglobulin receptor, or PIGR. So this is a protein on cells of the breast tissue inside the breast, where they bind, the antibodies will bind these proteins, and that directs them basically through the layer of cells between the mom's tissue and what will then on the other side be secretions, which is the milk. And so I'll illustrate this in a second. So the PIGR transports the antibodies into the milk, and then secretory component, which I showed you in that diagram before, gets attached by this process of transport. And it's actually a piece of the receptor. It gets marked. So when it travels through the cells to end up in the milk, the antibodies get labeled with secretory component. And this is really essential for the protection of secretory antibodies against enzymatic degradation in what is really a relatively harsh mucosal environment. So you imagine the baby's gut, it's acidic, right? Our stomachs are acidic. Our intestines are still relatively a little acidic. And nature had to evolve such that these antibodies could survive that harsh environment of the mouth, the digestive tract, right? So secretory component is extremely critical in protecting the antibody from being eaten up by the body's enzymes. So this is just illustrated here, just a bit of what I was saying. So you have your B cells, which are also known as plasma cells. They make the J chain, they make their IgA or their IgM. In a more minority of cases, you have the antibody being secreted in the mammary tissue, and then it goes through the gland and the cells, and it basically gets pumped through to, this would be the milk in this case. And then, this says, containment, right? So these antibodies then coat whatever they're meant to coat and protect against. And then here, a little bit more magnified, again, you see these dimeric antibodies made by these B cells that come from the gut. They bind onto the PIGR, and then they travel through the cells in what's known as an endosome. And then in that process, a piece of that receptor attaches to the antibody, right? You can see that. And then when it comes out the other side, this would be the milk, it's marked. And then again, you can see this is the whole molecule marked with the secretory component, and it's very important, and it protects the antibody from degradation. So antibodies have many different functions. I'm not going to get into all these functions today, but antibodies can, of course, block infection. So those I'll also refer to as neutralizing antibodies. So that would be when the variable region binds to its, virus in this case that it is meant to bind to, and that doesn't allow the virus to infect the cells anymore. So those are blocking. There's other functions that antibodies can mediate, and those would be more like if the variable region attached to the virus, but then the constant region of the antibody, like I mentioned before, it actually can, okay, one second. Can you still see my screen?
No, we can't see your slides at this point.
Sorry, it just switched to make me the host, so I'm not sure what happened. And I'm not able to share anymore. Oh, yeah, no, it changed my,
yeah, and I don't see. Bear with me a second.
I seem to have lost my ability to share.
And I don't see you on as. Sorry for these technical difficulties, everyone that's on. We're trying to get it worked out. Are you able, do you still have the link that I gave you, Rebecca?
Yeah, one second.
I believe you have to rejoin by clicking that link.
Sorry about that. One second.
Okay.
Hold on one second. You know what? It might actually be, sorry about that. It might actually be my own internet that has failed me.
You know what? Mine did too while you were starting this, so it must be something in the States because my internet dumped and I had to pull my other phone up to get on that. So do you have internet access at all right now?
Yeah, I think I've fixed the problem.
Okay, perfect.
I've now connected again. I'm connected.
Okay. Oh, there you are. Okay, hold on just a second. Let me give you the presenter mode again.
Sorry, everyone. Hazard.
Okay, you should be the presenter again.
Got it. All right, we're good.
We're back.
All right. Okay. So antibodies have many different functions. You good? You can hear me? Yes.
Yes.
Okay. So as I said, there's the blocking function, and then there's also many other functions mediated by antibodies, and that would be mediated through that other side of the antibody known as the constant region which actually can interact with a lot of different receptors on different immune cells. And those immune cells are present throughout the body, including in milk. So just to make you aware that an antibody that can bind to a virus, for example, may not purely just block the infection. It may not actually be able to block the entry of the virus to a cell. But what it can do is it can trigger other cells to attack viruses or attack infected cells. So there are many different functions of antibodies. And then, as I mentioned, there are many cells in human milk. So there's not just antibodies, and there are many components we don't understand, and there's many nutritional components as well. But there's also many different cells in human milk. So we know that there's, what we call the good bacteria, probiotic bacteria. There's a lot of that in milk. Those are cells. There's also human cells, the cells that are breast-derived, and there's immune cells that are blood-derived. So there's many different white blood cells in human milk, and I'm not going to get into this today at all, but I only show you this slide to make you aware that antibodies, as I said, can facilitate functions by many different immune cells. They can trigger all sorts of functions, particularly by macrophages, dendritic cells, neutrophils, and other types. And so milk is a living fluid, and it contains all of these cells as well when you're thinking about the immune components in milk. So now I'm going to switch gears a little bit, and I'm going to get to some background on COVID-19 just to familiarize everyone, and then how it relates to our work. So coronavirus disease 2019, or COVID-19, is an infectious disease caused by SARS CoV-2. That's the name of the virus. And that was first identified in December in China. And to date, there are over 13 million cases around the world and more than 570,000 deaths. So this virus is primarily spread between people during close contact via small droplets by coughing, sneezing, and talking. And transmission may also occur through even smaller droplets that can stay suspended in the air. So certainly transmission is a pretty easy thing. And between parents and a breastfed infant, certainly we would be concerned about just transmission from being near each other, not just through contact, but through, airborne droplets as well. And less commonly, you could touch a contaminated surface and touch your face, although that's like a less likely route of transmission. So SARS-CoV-2 is what we call a RNA virus. It's a positive sense RNA virus with one piece of RNA as its genome. And this is just an electron micrograph of what the virus really does look like, very, very magnified. And it's the 7th known coronavirus to infect people, which includes the most recent famous one, which was known as SARS. And each SARS-CoV-2 virus particle is, of course, very tiny. It's about 50 to 200 nanometers in diameter, so, of course, very small. And this is just a diagram of the virus. And I'm just going to talk a little about the proteins as we get at the immune response to this virus. We need to understand the proteins of the virus. So like other coronaviruses, there's 4 structural proteins. There's this little one that we call the envelope. There's the membrane protein. There's the nucleocapsid that wraps up the RNA. And very importantly, for the immune response, there's a big spike protein known as, it's the spikes of the virus, and it's known as the S protein. So this spike protein is responsible for allowing the virus to attach and fuse and enter the membrane of the host cell. So it's really critical for the function of infecting a cell. And it has a very strong affinity for a protein on human cells that you've probably heard of at this point called ACE2. And that's the receptor. So this receptor, what it does naturally is it's an enzyme, and it's attached to the cell membranes of many different cells in the lungs and arteries, heart, kidney, and intestines, and that's why the virus really can affect us in many different ways. So the viral spike is a highly important protein. As I said, it's the main target for a functional antibody binding, and this is illustrated down here. So you've got the spike on the surface of the virus, which has a membrane, which is actually the same membrane as we have on our cells, because viruses actually, when they grow, when they expand and they replicate, they use the cell membranes of our cells to create more of themselves. So they have the same membrane as our cells. Then we've got the spike, and that spike binds to ACE2, and that's a critical function. So antibodies that can get in the way of that are really important. So in children, COVID-19, we've all heard, it's typically, I say typically, much more mild compared to adults, which we can be thankful for. But, overall and this data, is ongoing, what we know about children and COVID-19, but about 10%, especially infants under 1, which is kind of the more critical age in terms of when you're looking at, the pediatric component of COVID-19 pathology, babies, about 10% of them who contract the virus will experience severe illness, which may not, in large numbers be deadly, but it definitely requires advanced care. So if you don't have that care for whatever reason, the mortality would definitely go up for this category. So given that COVID-19 pathology, we also know doesn't correlate with transmissibility, right? So we hear a lot about the asymptomatic carriers or pre-symptomatic carriers. So just because you're not coughing all over and you have a fever and all that doesn't mean that you're not infectious, right? So infants and young children are actually probably responsible for a lot of transmission, especially because you can't tell they're sick a lot of the time, but they might still infect each other or another adult. So that's, pretty concerning. And recently you've probably heard that in a minority, a small minority of children, they will experience something called multi-system inflammatory syndrome in children, or MIS-C. So that's associated with COVID-19. So after infection, there seems to be some kind of really concerning autoimmune response, most likely, that inflames the blood vessels, and this can be fatal. And it seems to only be occurring in children. So that is also very concerning. And so for all of these reasons, protecting this population, although, we think, COVID-19 doesn't affect kids, so is it a big deal? Well, it does, in the end. And so protecting this population is really essential. And one potential protective mechanism really might be, of course, the passive immunity by breastfeeding from a previously infected mother, or donor milk from a previously infected person, right? So this could be a way of protecting babies and young children. So right now, almost nothing, essentially nothing is known about the antibody response to human milk to SARS CoV-2. So some previous study of SARS was, first of all, pretty spotty when it came to anything to do with immunity in milk. Very little, very, very little data about SARS or MERS. And previous study, a little bit of placental transfer of antibody by someone who was pregnant when they were infected. Do the babies get that antibody to protect them? That would be transferred through the placenta. So that data was also not really clear, and there was some evidence maybe with SARS that placental transfer of these protective antibodies may not actually be happening, or it really depends on when the mom was infected during pregnancy. So then, young infants, newborns of mothers infected before or during pregnancy, then really need to rely on milk antibodies. You can't assume that they got it in utero. So much more research is definitely needed. It's very urgent to test for human milk for SARS-CoV-2 specific antibodies and their functions. And, knowing the types and degrees of COVID-19 antibody in human milk will inform policy about, breastfeeding and infection and treatment decisions for the scores of pregnant and breastfeeding people who will become infected by SARS-CoV-2 across the world. And of course, we have to keep in mind that any evidence of specific antibody in human milk, we have to weigh it against whatever risk there might be of vertical transmission of SARS-CoV-2 through human milk, which right now doesn't really look like we should be too worried about it, but a lot of that data, is being gathered now. So we don't know for sure that milk cannot carry, the virus, but so far it looks promising that it likely does not carry live virus. So should we find a high amount of SARS-CoV-2 antibody after infection in milk, remember, these antibodies are going to be almost entirely in this secretory form, which are meant to survive in a harsh mucosal environment. So when you think of potential COVID-19 therapeutics and you think of the respiratory involvement in the severe disease that a lot of people end up with COVID-19, you can think, what if we could harness the power of these special antibodies in milk and use them as a respiratory therapeutic? Because respiratory environment, harsh mucosal environment, not so different from where these antibodies are meant to be, and people have secretory antibody in their respiratory tract to begin with. So again, it's meant to be there. So could we use this as a treatment? So that's a part of my research as well. So we need to study this, of course. So towards that aim, we recruited as soon as we could, starting in late March. At this point, we have over 1,200 lactating participants in this study to look at SARS-CoV-2 antibodies in milk. And this includes, at this point, about just over 800 who have recovered from COVID-19. So the data that I'm going to show you here illustrate the preliminary findings regarding SARS-CoV-2 antibodies. Specifically, I look at IgA. I'll show you data on the other types of antibodies as well as secretory antibody, and that's very important. And this early study that I'm going to show you the data from is just of 15 people with proven or highly suspected COVID-19 infection, as well as looking at some control samples, of course, of milk obtained before the pandemic. So now I'm going to show you the data, which I'm sure you're interested to see all this background. So where's it all leading? So in this study, for what I'm going to show you, about 30 mils or 1 ounce of milk was obtained from these study participants. And participants, as I said, they either had laboratory confirmed COVID, although at the time when I was collecting these particular samples, tests were harder to come by. I'm based in New York City, so we got hit hard and fast very early on. And of course, testing was an issue, which now it's much easier to get tested. So not everyone had a laboratory confirmed case, but based on their circumstances, they were extremely suspicious that they had COVID-19. And milk was obtained about, usually in all of these samples, it was about 3 weeks, between 2 and 4 weeks after symptoms had resolved. So we weren't looking to study milk while these people were what you might call like actively infected. I wasn't collecting milk from them. I wasn't going to their house when they were sick. I was waiting at least 2 weeks till after they had really gotten better. And in these cases, the milk is about 3 or 4 weeks after they had been sick. So milk was pumped at these participants' homes. These samples were all obtained really really in the peak of our infection. So these samples were obtained essentially all in April of this year. And so it was really a no contact pickup. Everyone is in the study continues to freeze milk in their own homes and keep it for the study, and then we arrange shipment or pickup as needed of multiple samples at once, because this is a longitudinal study. So we are asking people to pump every 3 or 4 weeks, but this data is just on the first sample from a very small number of these participants. And of course, as I said, I used pre-pandemic negative control milk samples, which were obtained as part of other studies before December 2019. And all of the milk, when we get it and we thaw it, we centrifuge it at a pretty high speed, and then we take off the fat, we remove the cells from the milk, we clean it up basically, and then we save it in our freezers. It's just now skimmed acellular milk. We save it until we're ready to do our analysis. So the way that we measure antibodies in milk, in this case, we used a pretty old standard type of assay, a type of test that's used, universally, called an ELISA. This is the enzyme-linked immunosorbent assay. So most of you may have heard of this, but just as a little review, so it's a plate-based assay, which I'll show you what that means, and it's designed to detect and quantify soluble substances, such as the level of antibodies in milk that are specific for a particular protein, in this case, the spike protein on SARS-CoV-2. So the way you do this type of experiment, again, this is not something I invented, I just optimized the way we were doing it for use with milk. In this case, we put the spike protein, we glue it down to these special plastic plates and then you would add on the milk, and then if there's antibodies in the milk, it will stick to what's on the plate. If there's not, then those extra antibodies or whatever else is in the milk that doesn't stick to the spike protein will get washed away. Then you add on something called a secondary antibody, which in this case has nothing to do with the milk at all. You understand, it's not derived from the milk, but this is something you can purchase that recognizes human antibodies. So it would bind to any antibodies that have already bound to the spike protein. So you see it's like a stepwise experiment. And then these secondary antibodies have a special tag on it that with the right chemical treatment can glow in a way that a machine that picks up light can detect and quantify. So at Mount Sinai, one of the first FDA-approved ELISA tests, diagnostic tests for SARS-CoV-2 was developed by a different lab at Mount Sinai that we work with all the time. So we were able to use a lot of those same reagents and validate and optimize our assay for use with milk, which of course is not, milk is not blood. They are different, and we have to optimize the conditions to work with milk. So we did that first. And then, as I said, the plates we're using are coated with the full trimeric spike, or in some data I'm going to show you just a piece of the spike called the receptor binding domain, which is, as it sounds like, the piece of the spike that actually binds to ACE2. So our samples, we tested them in duplicate in 3 different experiments, in separate experiments measuring IgA, IgG, IgM, and secretory antibody. So when we look for secretory antibody, because remember that secretory chain is like an extra protein that glues down onto IgA or IgM, so we can pick up whether an antibody has secretory component on it, but we can't tell if that is IgA or IgM, right? So we have to then look at all the data together to think, is this actually secretory IgA or is it something else? So I'm going to show you what I mean when we get to the data. And then I'm also going to show you, you're going to see that we had 10 pre-pandemic control milk samples. And that, I had to analyze those first because those we say, okay, these should not be reacting with spike from SARS-CoV-2 because all this milk was obtained before the pandemic, right? So any activity we see is what we would call background or non-specific binding or, cross-reactivity, right? So you look at those first, and you determine mathematically what you're going to call a positive cutoff to tell you if your samples that you're really interested in, from COVID-19 recovered donors, are those actually positive for antibodies. So we have to form our positive cutoff, which is what we did. So, okay, the results. We found, which I was kind of surprised, pleasantly surprised, that all of the samples, this is a small number of samples, so I don't believe this will just be 100% of every sample we ever look at. Again, only 15 were looked at, but all of the samples we looked at of people who had recovered from COVID-19 contained significant levels of specific IgA that could bind to SARS-CoV-2 spike. So initially we looked at just IgA binding by ELISA to the full spike protein. And as you can see, again, what we found here, the dotted lines, these are all pre-pandemic controls. So we do have what I'll call low-level cross-reactivity, of antibodies that are present in milk all the time, regardless of COVID-19. And the dotted line here is the positive cutoff. So this is mathematically determined that anything above this dotted line, you would say, yes, this has very specific antibody for COVID-19. And as you can see quite clearly, all of these solid line samples are all the ones from the COVID-19 recovered people. They were all positive for IgA directed at spike. So that was nice, a little bit surprising, but great, right? And they have fairly robust levels of antibody, which you can see. This is diluting the milk, right? So as you dilute, you expect that the effect should drop off to 0, which it eventually does. But you have pretty strong levels, and obviously there's variation because it's people, lots of variation, right, in humans. So then when we looked specifically at binding to the receptor binding domain, again, this is the part of spike that binds to the ACE2 receptor. So we then found that 80% of those samples bound to the RBD region of the spike. So this, again, was really, good. This was good. So almost all of the ones that bind to spike are specifically binding to the region that would interrupt the binding, most likely between spike and ACE2. So that's good. And then that's what you see here. This is IgA in this part A of the figure. Part B of the figure is showing the secretory antibodies directed at spike, RBD, at the RBD region of spike. So you can see that actually also all of the samples that reacted to the receptor binding domain of spike for IgA also react in terms of whether they have secretory component. So this, of course, as we'll show you a little more confirmatory data, it tells us that most likely this IgA is almost entirely secretory IgA, which we would expect, but you can't just assume. You have to, find a way to really demonstrate that. So we also found that the IgA response to receptor binding domain of spike was certainly dominant, and it wasn't necessarily concurrent with a measurable IgG or IgM. Okay, so you can see here that unlike what I just showed you, here we're looking at IgG reactivity to the receptor binding domain. Here we're looking at IgM reactivity, and obviously far fewer of the samples are coming up positive for IgG or IgM. Now, this isn't altogether shocking because there's also way, way more IgA in milk in general. So, could be the sensitivity of our assay, but also it may just speak to a really robust IgA response, which maybe isn't unexpected because this is a mucosal virus, right? It affects the respiratory tract, it affects the gastrointestinal tract. You're going to get a lot of those B cells coming from those places and then making their way to milk, right? So milk from COVID-19 recovered donors exhibited much greater IgA, secretory antibody, and IgG binding against RBD. So what does this mean? So what we did here is we took all that data I just showed you. We only took the values of reactivity against the RBD for the undiluted milk. So I showed you before what happens when you dilute the milk and then the binding goes down to 0, which is what you'd expect as you dilute the milk more and more. Here we're just looking at milk in undiluted form, which is really how the baby gets it, right? So in my mind, that's always the most biologically relevant. What is undiluted milk doing? And you can see that when we group together the values of binding between the all of the COVID-19 samples, so this is all 15 samples averaged for their binding at undiluted against the RBD. This is for IgA, secretory component, and IgG even comes up as positive when you compare it to the values for undiluted milk for the negative controls. Then for IgM, we did not get a significant difference, although you can see that there's a trend that this does look pretty different, but statistically we do not get a value that is considered significant, and that's because there's a big variation here. But it is trending towards a difference in IgM. If we had more samples, maybe we would have gotten that statistical power. So IgA and secretory antibody OD values, which is the binding value for undiluted milk, were found to be highly correlated. So that is like that graph I showed you before, where all of the samples that were positive for IgA were also positive for secretory component. So then we actually did the statistical test to demonstrate that was true. IgG and IgM actually showed somewhat of a modest correlation as well, which, we're not sure what that means, but it may mean that they're originating from the same place and the same time and coming into the milk kind of at the, together, which makes some sense. So you can see here, this is the really critical part of the figure. You see how this forms really like a straight line. So there's a direct correlation between the IgA that binds to the receptor binding domain and the secretory component. And this is extremely significant and correlated. So that's good because that means really everything that's in there is probably secretory IgA specific for spike, which is what we want. And then here, this is the summary data for everything I just said. So this is the 15 samples. You can get a sense of if they were confirmed or suspected COVID-19. So it's kind of half and half. most of them were postpartum, and 2 were infected when they were pregnant in the last 6 weeks of pregnancy. Most were infected after their babies were born, sometimes long after, as you can see here, because you can see the number of months postpartum that they were when we got these samples. So that these babies ranged from a month old to 32 months old, right, up to, including toddlerhood, and we can see that, we don't have the numbers to tell you if there's any difference in terms of the stage of lactation and the levels of antibody here. We can't look at that with these numbers, but you can see we had a big range for this study. And then you can see summarized the reactivity against the spike. So again, all of these samples were positive for IgA. All of these samples were positive for secretory component. Our data shows us that it's likely that all the IgA was secretory IgA, which is what we would expect. And then some of the samples were also positive for IgM, some were also positive for IgG, and a few, 4 of them, were positive for all 4 types of antibodies. So in the last few minutes, I'm going to show you some extremely early data we have on the function of these antibodies for just a few samples. And all I'm going to talk about is the blocking function, the neutralization function, which is really what most people care about in terms of whether these antibodies are doing what we need them to do. So when we measure neutralizing activity, just keep in mind what we're talking about here. We have our virus. It needs to bind to ACE2 on a cell, right? If you have an antibody that can stop that, that's called a neutralizing antibody. Then you don't get entry of the cell. Simple, right? So when we study neutralization in vitro in the lab, what we would do is we would dilute and dilute and dilute more and more the milk or the antibodies that we can purify from the milk. And then we mix them with virus. So you can mix them with real SARS CoV-2 if you have the right safety conditions, which not many labs have. As you can imagine, you require a lot of safety to work with this virus. Or you can make something called a pseudovirus, which I'm not going to get deeply into, but many labs are using what we call pseudovirus, which means it's a different kind of harmless virus that you can express a protein from SARS CoV-2. So we are actually using a pseudovirus called VSV, but it has spike from SARS CoV-2 all over its surface, and it does not have its normal surface receptor. So it's like it's a fake SARS CoV-2. And there's a lot of precedent in using pseudoviruses in the HIV world, and a lot of people had already made SARS pseudoviruses. So when the time came, many labs immediately started making SARS CoV-2 pseudoviruses, and they're very useful to study neutralization. So when we do this experiment, again, we dilute the milk or the purified antibody, we add the pseudovirus, we mix them together, we actually do a spin which encourages infection because it pulls everything down to the bottom of the plate, and we put it in the incubator. We add the cells, and we put it in the incubator. And then basically, because of this special pseudovirus, what's especially good about it is that it can glow, it can luminesce when it's infected with these viruses. And I won't get into that technology, but that's how we can detect infection. Basically, the cells glow. And again, we have machines that can measure this glow and quantify it. So what we found is that most milk samples containing spike-specific antibodies exhibited neutralization activity that is likely mediated by IgA. And again, this is very early, so I only have a few samples of those 15, but it's headed in, a very good direction in terms of what we are hoping we will see. So here you can see I actually used milk in the experiment. So this is not purified antibody, this first panel. These are 3 of the highest level of antibody detected in our binding ELISA that I showed you before, these 3 COVID-19 recovered samples. You can see that they have a very high % of neutralization against this pseudovirus as compared to one pre-pandemic control that was used in this experiment. Now, when we purify the IgA, what we found, again, very few samples, so you can't make a big generalization, but what we found, which was interesting, was that 2 of these samples, again, this is just the IgA component, had very strong ability to block virus from infecting the cells. 1 really did not. And the pre-pandemic, you can see this is where this background activity comes in. So that means, 2 out of the 3 this neutralization activity is coming from IgA, most likely. One of them coming from somewhere else, some other component of the milk. I mean, milk is very rich in all sorts of interesting natural defenders against viruses, and we just don't understand this. And of course, this all has to be repeated with many more samples. When we looked at the IgG component, we actually don't see that IgG at the same concentration, really, as being used here, it's neutralized. So that was very interesting, too. So IgA seems really critical here, which was, again, a feature of milk that we kind of expected, but I also might have expected to see the IgG doing some neutralizing, which it did not. And then just one other experiment we, again, ran. So this is with milk, again, not purified antibody. This is with milk. Again, repeating some of these samples, which again demonstrated good neutralization. And then we added in a few more samples that were obtained before the pandemic. So what you can see is that 3 out of the 4 COVID-19 samples here had very strong ability to block virus from infecting cells. And then we can see that the pre-pandemic samples, some had background where they could do it undiluted. Some really couldn't even do it at all, but it's very different than the samples obtained in 3 out of 4 cases from COVID-19 donors. So this is definitely very promising preliminary data in terms of neutralization, and that's what we're really working on right now. So now I'll just summarize for a couple minutes, and then we can do some questions. So all of the milk samples we studied were positive for spike-reactive antibody of at least one subclass, which was IgA. And I want you to understand this is just a snapshot of a dynamic immune response. So we don't know what it's going to look like in 3 months. We don't know what it's going to look like in a year. And this is, very few people. So it's not just a one-time test. We have to keep looking. More samples follow up, and that's what we're doing now to really understand this response. So, this is unprecedented, but as I said, if this response is strong, we could extract these antibodies, which will be primarily secretory IgA, and use them therapeutically for anybody, not just babies, but these could be applied therapeutically if they're very, at strong high levels, which they appear to be in most people. So we didn't compare these milk donors' blood antibodies to their milk data, which is something I'd like to do in the future. But it was evident that a lot of samples contained reactive IgA without containing measurable antibodies more associated with serum, like IgG. So, we don't know why this was. It could be that they're so much lower in milk we couldn't detect them, or it could be actually there's a much better IgA response in general, which would not be totally surprising. So all samples exhibited positive IgA. They also had positive secretory reactivity. And this suggests that pretty much all of the IgA was probably secretory IgA, which is very relevant to using this as a COVID-19 therapy because, the basis of that is that secretory IgA is very unique from the IgG therapy, which would be intravenously infused and derived from convalescent plasma, which you've probably heard of, and the secretory IgA is very durable. So probably we would need, effectively a much lower dose. If we gave it by respiratory route, it would be highly directed and highly functional in the respiratory tract if we took it from the milk and used it that way. So it would not be difficult to source this milk from COVID-19 recovered donors. There are so many people infected globally. Milk can be given every day. You can increase the milk supply with pumping, as we all know. And it's not unrealistic in my mind that donors that were inspired to do so could give us, 100 mils or even 200 mils every day. Many people, once breastfeeding is established, could do that if they were inspired to donate in that way, if they knew that the milk was being used for this purpose. So we could get, even from 100 donors, we could get as much as, I estimate, about 6,000 milligrams of secretory IgA every day. And that's only from 100 donors, right? And we're talking global donors here. So I think this is actually a really viable potential treatment that I'm exploring and trying to gather data on with the hopes of being able to test this as a therapy that would complement other therapies such as, infusion of intravenous antibody and other antivirals being tested. I think it could be part of a combination therapy. So as we keep studying, we aim to determine the efficacy of convalescent milk, the treatment for COVID-19, and utility of these antibodies to prevent or mitigate infant SARS-CoV-2 infection as well. And so this will have a lot of implications, beyond the pandemic, which will really fill large knowledge gaps in general regarding human milk immunology, which really has been highly understudied. So I'd like to thank everyone at the Icahn School of Medicine and my lab, especially my technician, Alyssa, my mentor, Susan, and also the Kramer lab, which gave us the proteins used in these experiments. I'd also like to thank UC Merced. I have some very important research coordinator and materials support coming from that institute. And of course, the milk donors, we couldn't do this without them. So thank you very much.
Okay, thank you so much for that information, Rebecca. That was very informative and interesting. We do have about 5 minutes for questions. If you want to submit your questions via the chat function, we will try to get to those. As we're waiting for questions to come in, certificates for those attendees here in the U.S. will be available tomorrow by logging into your account. And we do have a couple webinars coming up, and for more information on those and to register, you can visit www.medillaeducation.com, and that would be for the July 21st webinar and the August 19th webinar. We have a question that came in. It is, did you look at the milk of different, or I'm sorry, let me repeat that. Did you look at the milk of people infected early in pregnancy or late in pregnancy?
So no, we haven't been able to do that just because, this was actually really early on. So we have some people in the cohort now where they were infected, during pregnancy earlier, and now they've had their babies. And then we can, they would like to give their milk, and we will study that. So yeah, we weren't able to do that yet, but we will.
Okay. The next question is, since babies are possibly getting antibodies for SARS-CoV-2 from their mother, would you recommend keeping mom and baby together post-delivery? I've seen some stories about hospitals separating positive or presumed positive moms from their babies.
Yes, I absolutely recommend keeping mothers and babies together, regardless of, who is testing positive, whether it's the mom or just the mom or the mom and the baby. The benefits of having them together far outweigh this tiny, tiny potential of, a transmission situation that, especially globally, separating the mom and baby could be deadly for many reasons. Breastfeeding needs to be established. In America, yes, there are other ways to feed the baby, and that's all well and good, but it should be up to the mom and the family to feel like whether they want to be separated or not, whether, establishing breastfeeding is important to them or whether, that wasn't something that they were, banking on, so that's their choice. But in general, it should absolutely not be a mandate to separate these moms and babies because there's pretty much no evidence that there's transmissible virus in the milk. Mom can wear a mask, mom can wash hands if people are really concerned. And, it certainly looks like that she's also giving very important antibodies to the baby as well.
Okay, thank you. We're almost at the top of the hour, and I don't see any other questions coming in. Again, Rebecca, thank you so much for delivering this presentation and the information for us today. Thank you, everyone, for joining us and taking the time, and enjoy the rest of your day.
Thank you. Thanks for having me.
You're welcome.